Hi, I'm Sofia. I work at the Cardiovascular Research Institute here at UCSF. So what we're doing right now is we are conducting a PCR, which is a method we use to take certain proteins and kind of mishmash them together to create this code that we can decipher. And like what we're doing now is that we're getting these tubes, and to conduct the PCR we are including some water, some PBS, which is basically just another acid which we put to that helps condense the DNA and like the contents, like the actual parts of the chikai itself. So that's the water. So I like code this on a computer, and yeah, that's it.
Hi, my name is Erika, and I've just taken time points of my React experiment, and now I want to put in some lemon buffer to mix into my experiment to make sure the reaction stops, and that way I can boil that down and put it into a gel. And so I'm putting 5 ml. So I'm just mixing the solution around to make sure the buffer mixes with my Okay, so now that I finished boiling them, I need to spin them down. So now I finished spinning them down, and I'm going to put them into a gel and run it to see my results and see where my protein is.
Hi, my name's Lina. So today I'm gonna put some media in these well-played salts. So first we're gonna use ethanol to sterilize. So I'm gonna add 200 microliter of media.
Okay, so
So now I'm gonna put these back into the hood and we're done.
Hi, my name is Norman David Lopez, and this summer I was at the Macau lab at Mission Bay, and I worked with chick embryos. And the lab's big question is, how is bilateral symmetry patterned and achieved throughout development? And my question for the summer was, does ECM proteins act as a structural barrier? And just a little background, ECM, it's an area outside the cell that contains certain proteins like laminin and fibronectins, which I looked out specifically, and we want to test whether or not it worked as a structural barrier that prevents cell migration. And if you see in the picture right here, during a process of we characterized the midline pyramid streak using immunohistochemistry, and this is what we got. So we use blue for the nuclei, DAPI, the laminate, we use actin for laminate, and we use red for our first antibody for ECM. And as we can see, laminate is highly enriched, and the pyramid streak midline is just this area. And you can You can clearly see that there's a strong presence, and you can really appreciate it. I'm sorry, you can stop it right now if you want. Ah, there it is. And you can clearly see this is a small area of the permanent streak, so it'd be just this and not the whole permanent streak itself. We took a picture at a higher magnification, and you can see here that laminate is clearly present in the permanent streak, and here at the transverse cut you can see that there's a few cells in the permanent streak itself, but the majority stay at the respective side. And this clearly gives us an image that ECM is present, and it does work as a structural barrier. And in order to test whether or not it worked as a structural barrier, what I did was in vitro gain of function, and we introduced an exogenous wall. Unfortunately, during this process you have to be very precise, because as you can see in this movie I'll show you, if you're not as precise, your embryo will shrink like it did here under the microscope. If you leave too little liquid, then it's going to shrink like it did here. If you leave too much, then your embryo will float away and you won't get good images. But what we expect to see, as normally our cells would migrate towards the permanent streak, represented as this tan line, and they'd bounce off and go back to their ipsilateral side. But if we introduce our exogenous fibronectin wall, then the cells would move as normally, but once they hit the wall, they'll bounce off of it and go to the permanent streak. And if that happens, then my hypothesis is correct, and the ECM proteins act as a structural barrier. But if the cells migrate towards the exogenous wall and they ignore it, then my hypothesis is incorrect, and there's something else in the midline permanent streak working as a structural barrier. And that was my project for the summer.